materials for you, study materials on Canvas, which I'll come back to in a second. And then, so the majority of your practical, well, the practical is on this key from Lab 26. If you haven't handed in Lab 26, I will grade it right away and get it back to you today so that you have it. So what you need to know is, I'm going to circle the things you have to know on your own. So those are the things you have to know. If you want to take a picture, you can go ahead and take a picture. You have to be able to write these words out. You're not going to get a word bank. So I don't think it's terribly too much. I'll have specimens. I'll have 20 specimens out. A lot of them will be directly from this lab. There would be a few that you haven't seen before that you'll just have to be able to, and they should be pretty easy. Like, for example, the ones that I'm probably going to have out that you haven't seen before will be some kind of flowering plant, and then you'll have to identify, are they a monocot or a dicot? So, things that you really need to focus on is just knowing by look what these things are. So fungi can be anything from Lab 27. So today in the lab, all the different fungus that you're going to look at, I could have one of those specimens out, and the question will be, what is the kingdom? And you just say it's fungi. There'll probably be about 5 or 6 of those. Those should be real easy because you don't have phylum and you don't have a class. So anything in Lab 27, that's all you gotta know. They're fungi. They'll be the class fungi. I mean the kingdom fungi. The rest of these, chlorophyta, here's the main things. They're going to be green, and they're going to be submerged in water. My grading is really terrible. So they're going to be green and submerged in water. If it's not a fungus or a protist, phylum Chlorophyta, if it's not this, they're going to be a plant. So everything down here is kingdom plantae. I will ask at least one question where I just put a plant out and say, what's the kingdom? So that should be nice and easy. Plant, kingdom plantae. The bryophyta plants, these are really, really short. They will be growing on a log like a piece of wood. So short, fuzzy, green. So the bryophytas. Short, fuzzy, green, they're growing on a log. Pteridophyta. Pteridophyta are going to be ferns. And the ferns have pinnate leaves. when you're talking about pinnate leaves, it's like the leaves have leaves. So if this is considered a leaf, then it has little leaves on it. That's what we mean by pinnate. Ferns, other things that ferns can have. I don't think we have any spores. I'm not even going to put that down. And I don't think we have any of the fuzzy roots. So this is really what you need to focus on. Like I know none of our the other characteristics that identify something as a fern is that they have spores on the undersurface of their leaves, but I know we don't have any that are making spores right now. And then they also can have those like fuzzy stems that come out that look like spider legs. I know we don't have any of those either. So I'm not going to distract you with those, but just know you've got these pinnate leaves. Gymnosperms. Needle-like leaves. So long, skinny, very, very skinny. We're talking about like, look, I made it so skinny you can't even see it. Like little lines. Needle-like leaves and cones. Like pine cones. So that's the gymnosperms. So if you see something that has long, very, very skinny needle-like leaves, or you see pine cones or both, that's a gymnosperm. The angiosperms have flowers. Not everything's gonna have a flower. I'll put flowers. I'll make sure they have a flower. That'll be a good clue. So yeah, flowers. If it has a flower, but remember some of the flowers are weird, like grasses. Remember the grasses? They have those like little spiky flowers. So some of the flowers, just make sure you go over, and I'll have it in the practice today. Make sure you know like some of the flowers are weird. They're not like just, bright, colorful. This is probably the weirdest one, is that one. Flowers, and here's the other thing. Seeds. If I just put out a whole bunch of seeds, that's an angiosperm. Monocots, here's key things. Parallel veined leaves, flower parts in threes, and one cotyledon, which means the seeds are one piece. The dicots, net veined leaves, flower parts in 4 or 5, seeds, 2 pieces. 2 cotyledons. You can just put angiosperm on the test, monocot and dicot. Okay, so maybe not. Well, my amazing notes. But those are the things you have to know. You have to know that. I don't think it's a lot. So that's what you gotta know. You have to know these names. You have to be able to spell them out. Does spelling count? I mean, you gotta be really, 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 really close. Like I've had people put like gymnostar. I'm like, no, that's not close enough. So get close. Like if you put gymnosperm A, like that, that's fine. But it's gotta be, again, very close. And Anybody questions?
So it's no multiple choice?
No, it's just, you'll walk up to a specimen and the card will say kingdom, question mark, phylum, question mark, or class, question mark. So you don't have to put down if it says class, you don't have to put plantae, angiosperm, dicot. You just put dicot. So whatever it's asking for, that's all you have to put down. Anybody else? It's a nice practical to start with because not a ton of information, and it should be easy as long as you study. But that's kind of life, right? This isn't. I would say this is very low yield, big yield for low stakes. You don't have to put a ton into it. So let's take a look at where you will find your study material. So here's our class, and if I scroll down, okay, so our unit here, evolution and diversity, scroll down, at the bottom you've got lab practical on labs 26 and 27, study materials. I borrowed this from Professor Bernard. It's basically kind of goes through a lot of what I did in the lab last week, a little more detail than I just went through. So that's a nice kind of lecture review. And then you've got these to go through. So here you can see the leaves. This one has a lot of spores, but like I said, we won't have a lot of spores. I don't think any of them do. So the leaf has leaves on it, leaflets. So this is a pinnate leaf. Because the main leaf, the whole leaf, has a lot of little leaves on it. So that's good fern. Monocot or dicot? Which one? 1, 2, 3, 4, 5, 6. So that's a monocot. What is the phylum? It's got needle like leaves and pine cones. Yeah, gymnospermic. Good. Trees are angiosperms. Is this a monocot or a dicot?
Monocot.
Which one? Dicot. Okay, so main vein, and then the other veins radiate off in a net-like pattern. It's kind of a hand-shaped leaf. These are seeds. 1, 2 parts. Hat and body, 2 parts. Dicot. It's a phylum. Short, fuzzy. This one's growing out of rock instead of wood.
Bryophyta.
Bryophyta, excellent. Bryophyta. Submerged in water in green. What's the kingdom?
Protista.
Good. Protista. What is the phylum? It's with a C.
Chlorophyta.
Good. Chlorophyta. See these right here? Those are flowers. Those are cornflowers. So you can see, if you can see, the veins are all going like this in the same direction. Monocot or dicot? Good, that's a monocot. Good. Mushroom.
Fungi.
Fungi, that's good. Grass, see all these little flowers here? The tops are flowers. The veins are all going in the same direction. Monocot or dicot? Monocot. Okay. Couple of features here. Veins in the same direction. 1, 2, 3, 4, 5, 6 petals. 1, 2, 3, 4, 5, 6 stamens. Carpel is in a group of 3. Monocot, right? Multiples of 3 and veins going in the same direction. here we have an oval leaf with one vein down the middle, all the other veins coming off. 1, 2, 3, 4, 5. Good, dicot. Okay, so just there's other study materials for you. Hopefully you'll feel like just going through that not too bad, right? So use the study materials. You got this. So in lab, I will do a little practice one. 2 lectures today, 2 sections, I should say. Protists and Prokaryotes. So let's go to those. Protists on page 85. Protists, there is so much research going on in this field. There's so much to do. There's a lot of exciting work going on. The protists used to be one kingdom, and then people started going, wait a minute, these protists are so incredibly diverse. So let's talk about why they turned them into kingdoms and not a kingdom anymore. Still, again, things are being worked on. Many are unicellular. Uni means one cell. But some of them are unicellular that live in a group. So it's like, you know, a bunch of humans living in a house together. It's kind of like what they do. They live together in a group called colonial. And then some of them are bigger than one cell, and they are multicellular. So there's a good diversity in the amount of cells that some of the protists have. Some of them are autotrophs. Auto means self. Troph means feeder. They feed themselves inside of their cells, which means that they typically photosynthesize. Occasionally you have some that do chemiosynthesis, which is they do like a chemical process like photosynthesis because they don't have sun. And then many also are heterotrophs, other feeders. Hetero means other, troph means feeder. They have to get their energy from somewhere other than their own cells. Most of them just reproduce asexually. They make a genetically identical copy of themselves. They don't use sperm and egg, but some of them do make sperm and egg. So we've got just like some basic features, we've got a lot of diversity, and there's a lot more ways that we can like diversify them. So we're going to talk also about how some are plant-like, some are animal-like, and some are fungal-like. We'll put them into those categories too. So some of them look like this, some of them look like that. They move a lot. You probably did a lab in like 3rd grade where you looked at a whole bunch of different kinds of protists swimming underneath the microscope. You can see this one, for example, has chloroplasts inside of it, so it is autotrophic, makes its own food. But it's different than a plant where plants are kind of stationary. These can swim around. You can see all these cilia. There's hairs, and they just like, they go fast. So we'll start with the plant-like protists since we took a look at that one. The diatoms. Diatoms are also called phytoplankton. There's phyto for photosynthesis. They're autotrophs, like plants. Plankton, this term, just an FYI, is plankton means anything that lives in water that cannot swim against a current. So that if a current comes, they just are like, woo, and they go. Most phytoplankton are microscopic, but jellyfish, for example, are phytoplankton. Because jellyfish are just like, woo, woo, woo. If a current comes, they're just like, woo, they go. So sometimes there's big plankton. Tin, algae, tin? Oh, I think that's, huh? What do you think I meant by that? I don't know. What does it say in yours? Oh, tiny. Oh, I was like, thank you. Oh. I was like, what do I mean by tin? Tiny, they're tiny. They're tiny, a lot of them are microscopic. These are underneath the microscope. Diatoms, as I mentioned, are autotrophs, and they're photosynthetic then, and they're unicellular. You can see here. A lot of them have silica shells. silica is what makes glass. And they come in like all kinds of cool geometric shapes. They're really beautiful to look at. Check this out. This is, I think, the wildest thing about them. These microscopic photosynthetic organisms provide us with at least 70% of our oxygen. So if you're thinking about like warming of the oceans and climate change, I'm thinking like, I live in Illinois, I don't live near an ocean, I don't have any relationship. We absolutely all have a huge relationship between the oceans, and especially these producers in the ocean, or these autotrophs. They provide us with the majority of our oxygen. So the warming of the oceans is critical to humans in terms of slowing down climate change. They are also the core, the producers in the ocean food web, food chain, trophic levels. We'll get to all that at the end of the semester, but they're what everybody else eats. That you have groups that eat them, and then groups that eat them and other animals, and then groups that eat them who eat them who eat them. So they're also like our plants on land that produce food for us. They're like that in the oceans. So the warming of the oceans could cause them to collapse. It causes food to be gone for the ocean ecosystem, and then also we lose all that oxygen. There are a lot of different kinds of algae. So another kind of algae are the seaweeds. There's a bunch of different kinds of seaweed. This one's called brown seaweed. This is found in colder waters. This is giant kelp. Giant kelp are pretty much like trees on land. They get very, very tall. They have these like funny holes onto rocks on the bottom, and then they can grow hundreds of feet tall. So like in here, when you're looking, it almost looks like a forest, but with water. And all of these organisms that you see, and then there'll be a lot at the base, also a lot of invertebrates that live on the ground. They all, even within these long strands, there's a lot of animals and other organisms that live within there. So they provide a lot of homes, just like trees do. They give shading. They are the producers. So this provides food for a lot of animals as well. This is red algae. Red algae is interesting in that it can take calcium out of water and it can incorporate it into its body. So I know like maybe when you were younger, everybody said, oh, you gotta drink cow milk to get your calcium, but calcium, you can get it in so many different ways, like green leafy plants. They also do the same thing. They incorporate calcium into the leaves of their body. It's not, right, if you eat spinach, it doesn't taste like, it's not like crunching on a bone. It's just like a softer calcium, but it's the same thing with these here. We actually find red algae in a lot of products. Any of you have your red algae today? I hope so. I hope y'all brushed your teeth. Usually red algae is found in toothpaste. You'll see this term carrageenan. A lot of gooey stuff, toothpaste, like chocolate milk, whipped creams, ice cream. A lot of beauty products have carrageenan in them. So we probably all, I hope, use them today. And then in freshwater ecosystems, we have green algae. Green algae is thought to be thought to have a common ancestor with land plants. So when everything lived in the ocean and then plants had mutations to make their way onto land, they believe there was a common ancestor with the green algae and land plants. This is just from our pond out there. All right, so here's some cool things that are going on in terms of technology with this kind of protist. They're using green algae for biofuel. This is my challenge to learn you. Go into this field of research and try and figure out how to grow this green algae, yet keep the cost of like electricity down for running your farm, because the cost of the electricity that you're using to run the farm and the amount of energy that you're getting out of your green algae, it's like this. It's just like a little bit more cost to run your farm than what you produce. So there's a lot of research going on that they're trying to get it so it's like this, right? So that we get less energy put in and we get more yield out. If you figure this out, you'll be so rich, because this is what we need. However, one of the things about something like this, like these algae farms, so one point that's really important here is that these kind of algae, they can triple their weight, double to triple their weight in a day. That's amazing, right? Can you like even get close to doubling your weight in a day? No, not even close. So that's one of the things that makes them a really good candidate for biofuel, is that there's all kinds of different biofuel research going on, like corn is one thing that's being used, switchgrasses. That corn, you can get one growing season a year, right, out of corn, but these get a growing season every day or multiple times a day. So it's a lot more yield and biomass for this kind of fuel. Trying to think of what that is. One other thing. These can be grown not only, like this one you can see is outdoor, that it's like giant test tubes, right? But in the Midwest, you could set something up like this. This is an indoor biofuel, so that you can have like a big glass house and you can grow these inside. So this kind of research can be done anywhere. You just gotta figure out that energy, get that energy up. The other thing that's really important about this, regardless if the energy yield is less than what you're putting in, this is renewable because you can keep growing it, right? They are going to replicate themselves 2 or 3 times a day. So that's one of the things that's really good about it. It's one of the, I don't like to talk, I mean, I do like to talk politics, but not in my classes necessarily, but right now there's like a big push to go back to fossil fuels. But a lot of the companies that invest in fossil fuels are also investing in technologies like this, and they know that this, in the long run, right, fossil fuels are going to run out. They're not limitless, but these, you can keep making and making and making and growing them. So the longevity of this kind of fuel is somewhat limitless. So a lot of people, while there's being this call to go back to the U.S. focusing on fossil fuels, a lot of the fossil fuel companies are like, wait, let's think about this. I'm investing a lot in this too. So we'll see what happens. But definitely, like, really exciting stuff. There's another kind of protist called the dinoflagellate. They kind of look like dinosaurs. That's where they get the dino from. They're microscopic. They're found in the oceans. One species or one group of these dinoflagellates, they produce these red tides. You often hear about this in the Gulf of Mexico, usually in the summer when it gets really hot. And it gets like kind of goopy and swampy at the ocean. So one thing is for ecotourism, it's not good. Because would you swim in this if you came out and you got all this like slimy stuff all over you? Probably not. But the other thing is that this causes irritation to your skin as well. So for ecotourism, it's bad. The irritation to our skin is caused by a toxin that this kind of protist produces. And not only is it just like cause irritation to our skin, causes irritation to the skin of any organisms living there, but also poisons them. And so if you are somebody who has a business of like producing shrimp or some kind of fish that you sell, and that's your livelihood, when these, they call them red tides because they're red, and that produce those toxins, you can't sell any food, any shrimp or lobsters or fish or clams or crabs, whatever your livelihood depends on, when these red tides occur. And we're seeing these red tides occur more often and in greater amounts due to climate change because of the warming. They do great when it's warm water. Another kind of dinoflagellate, they're called zooxanthellae, so it's like zoo. Some people call it zooxanthellae, but zooxanthellae. These are found underneath the skin of corals, and they give corals their beautiful colors. I know these look kind of brownish here, but that will give some of the corals really, really amazing colors. And they are in a symbiotic relationship that the corals get about, 90% of their energy needs from this kind of autotrophic protist, and so then they can just kind of like be pretty and do their thing. Okay, so those are all the autotrophic protists. Let's talk about the animal-like protists. These ones move. That's why they're animal-like. They have a lot of movement to them. They're heterotrophs. They have to get their food from somewhere other than their own cells. They'll eat the autotrophs. There's a big group called Paramecium. There's a lot of different species of Paramecium, but also include Plasmodium, which causes malaria, amoebas, and then zooplankton. So zooplankton are going to be anything that lives in the water that is animal or animal-like, and they eat the phytoplankton, and they get pushed with the currents as well. So kind of cool that you have this whole like microscopic ecosystem on the top of the ocean going on. I always, my marine biology class, we always take like a seine, which skims the top of the water, and then we look at it under the microscope, and then we get to see this whole ecosystem under the microscope. And so, some of these, so kind of, I think, interesting, let me get to amoeba, yeah, correct. So amoebas, there's a really, really old film called The Blob. Like, look it up on YouTube at some point. It's, the whole idea was this, the idea of it came from the person who wrote the film from seeing amoebas under the microscope. They're larger, like you can see them on a slide, you can see them with your eye, they're small, but then you can see the details better underneath the microscope. And what amoebas do is they make these what are called pseudopods, which are like arms, and they can just, imagine that you could just like project out this blob thing that could grasp something and pull it into your body. And so these pseudopods can just go like, and pull something inside, capture something. And that is the idea of the Blob movie. But they made this amoeba-like organism, they made it life-size, so that the blob was like going around and getting people, and that's where it came from. A lot of these protists, they're terrifying if you thought about them in terms of being human size. But they have this whole crazy ecosystem that has their own little like crazy web of eating each other. Amoebas, some amoebas are like no big deal. They just are around, they do their thing. But some of them, so they are predators. So here's kind of just another interesting thing to think about, that these microscopic communities in water, they have predators, they have prey, they have like symbiotic relationships. They have all the things, all the relationships that we do on land, they do all that at a microscopic level. So amoebas are predators. They go after and eat other organisms. Some of them are parasitic. So if you've ever gone to like a foreign country and you get that, you know, like your stomach not good and you end up like puking and having diarrhea, might have been caused by an amoeba. There's a lot of other things that can cause that, but it's called amoebic dysentery. And then occasionally we hear about like somebody went swimming in a lake and they died 3 days later because they got a brain-eating amoeba from swimming in that lake. Another kind of animal-like protists are Plasmodium. Plasmodium are a parasite that cause malaria. This is another interesting thing. they have a host. They have two hosts. They have a primary and a secondary host. Primary host is the mosquito. They infect a mosquito. They have these microscopic spores that they produce. And then the mosquito, when a mosquito stings a human or another organism, the stinger, it basically injects this into the human. And then in our bodies, what happens is we get malaria. The mosquito isn't affected by it. They don't die from it, but we do. They'll get into the liver and the blood, and then they get onto our red blood cells. And our red blood cells make up about 60% of our blood itself. They are 99% of our blood cells. They cause your red blood cells to rupture. Our red blood cells are really, really important for our survival because they carry oxygen, and carrying oxygen helps us to efficiently break down our food. If you remember from evolution, talking about aerobic respiration, is that this really, having malaria messes with your ability to do aerobic respiration or break down your food. And when they get in the blood cells and reproduce, they make lots of spores, and then those spores infect more blood cells and more blood cells and more blood cells. So they can cause fevers. They'll the big thing about malaria are the high fevers, and then infecting your smaller areas of your body, like nose, ears, fingers, toes. So some people, if the infection goes a little bit too long before treatment, they might lose like a finger. It is curable, so that's a good thing, is that you can get cured. So this just shows you that the mosquito gets these spores and carries the spores inside of them, and then when they bite you, they inject those spores in your body, which make a home in the liver, and then the spores reproduce and then get into the red blood cells and cause red blood cells to burst, and then slow down your ability to send oxygen out, especially, like, most importantly, to the tips, the smallest parts of your body. And when those body parts basically starve from not having enough oxygen, then those things can die off. All right, last group, fungal-like protists. They kind of look like fungi. So if you remember from last week, the structure of fungus looked like this. It had hyphae or hyphae, strands. You can see them underneath the microscope today. And then it also had spores, and it kind of looks like this. So for a long time, a lot of these fungal-like protists, they thought they were fungus themselves. And now they realize that they're not. They're structurally similar. So here we have homologous structures, structurally similar, but not closely related. Many of them are decomposers. So this has been very detrimental to vineyards if they get infected, avocado trees, and potatoes. The Great Irish potato famine was caused by one of these kinds of protists. So just a little taste into the protist. Let's talk about prokaryotes. So we've been going like smaller and smaller and smaller and smaller. So we get to the prokaryotes. The prokaryotes have two domains, the domains bacteria and archaea. There's only three domains. Remember that there's the bacteria, the archaea, and then there's the eukarya, and anything in eukarya has a nucleus. Up until this point, we've been talking about organisms that are in eukarya. So now let's go to we're just gonna kind of lump them into one big lecture. We'll talk a little bit about specificity in a bit. So let's just go over these terms again, prokaryotic and eukaryotic. Pro means before, karyotic means nucleus, eu means true, karyotic means nucleus. Prokaryotic cells evolved before the nucleus evolved. They are believed to be our common ancestor amongst all organisms on the earth. They were around for about 1 million years by themselves with no other organisms. I mean, there are a lot of different kinds of prokaryotes, but there were none that were eukaryotic. Prokaryotic cells are typically simple. They don't have a ton of different kinds of organelles. They don't always have organelles that have membranes. There's a lot just more simplicity, but they've been around for billions of years. So sometimes when we're talking evolution, being complex does not mean better necessarily. They have a lot of features that allow them to survive in crazy environments that we certainly could not. Eukaryotic cells have a nucleus. Eu means true, karyotic means nucleus. You could see this eukaryotic is an adjective. It's a eukaryotic cell. This describes this noun. Or we could say eukaryote, which is like one way of just saying a eukaryotic cell. So just know, I know sometimes students are like, what's the difference between eukaryotic and eukaryote? Eukaryote's a noun. Eukaryotic is a descriptive term or an adjective. Eukaryotic cells have a nucleus. They have membrane-bound organelles, which allows their organelles to filter what comes in and out of the organelles. So there's a lot more control in that kind of cell, a lot more complex. Here's our phylogenetic tree. I would guess that you might see this again somewhere. The bacteria and the archaea, why they have a branch here means that there's a common ancestor between the two of them. Remember that when we're looking at phylogenetic trees, that every time you have a branch, it shows a common ancestry. This is the common ancestor down here. The common ancestor. And then that common ancestor, right, natural selection happens. The population goes into different areas. The force of natural selection acts on them differently, and then they start to, based on their mutations that they have, they start to have different features that are favored, and they start to become different organisms, or we have the process of speciation occurs. The archaea have a common ancestor with the eukarya. And then we have the eukarya, where our closest ancestors are the fungi. So remember that the clades, my reminder again, the clades show common ancestry at those branches. So I stuck this again in here from the beginning of talking about diversity again. So maybe you should really study that. All right, there's 3 common shapes. So there's certainly more diversity in this, but we like to just, this is general overview class. So 3 common shapes. 1 are spherical or round. We call them also cocci. There's rod-shaped bacteria. Sorry, I went fast for that. I missed it. Cocci are the round bacteria, spheres, spherical. The rod-shaped bacteria, those are called also bacilli. And then another common shape are the sporilla, or spiral or corkscrew shaped, kind of like DNA, like a helix. Even though it looks like a snake here, it's like this. If we could see it in 3 dimensions. Anything that has like a curve to it are these sporilla bacteria. And then again, you can see cocci, bacilli, sporilla. This one you can see a little bit more of that 3-dimensional shape to it. Sizes of these cells, they're tiny. So let's take a look at, this is, for example, a eukaryotic cell. And we're working in micrometers, so these are tiny, tiny, tiny. But let's take a look at like a cocci here. Look at the relative size, really, really small. Some of them are quite big, like a cyanobacteria, which can photosynthesize. They're not like terribly too small. So you do have a lot of diversity in sizes. E. coli, you can see, is a smaller kind of bacteria. And just to give you just a little like idea, we don't get too much into viruses because they're not living things, but these are virus particles here. So virus particles are really, really busy and tiny, and especially in comparison to our cells. So just to give you some relative ideas. Okay, which of the, oh no, sorry, I went too fast. Which of the environments would you find prokaryotes, right? Everywhere. We're gonna find them everywhere. Okay, how they reproduce is by fission. Fission means to split into two. This I think is a pretty amazing way of asexual reproduction, is that they can, in about 20 minutes, some of them, 20 minutes, they can grow to twice their size, copy their genetic material, all of their organelles, and whoop, split into two. 20 minutes. That's why sometimes when you wake up in the morning, you have something in your throat, you have that day where you wake up and you're just like, oh no, and then the day goes on and you start to feel worse and worse and worse. It's because they're just, they're reproducing, they're doing fission inside of you, making you feel worse. This is the simplest form of reproduction, asexual reproduction. They make a genetically identical copy. Granted, we also know that in copying of the genetic material quickly allows for high mutational rates, which allows for quick evolution. So thinking about the bacterial lab that we were looking at antibiotic resistance, the way that the bacteria evolved antibiotic resistance is really due to this, is their asexual reproduction, is that having the ability to quickly reproduce and copy your genetic material allows for a great deal of mistakes to slip on by, and they can get all of these different kinds of antibiotic resistance. So a big thing is that because of their quick ability to reproduce, they can evolve also very quickly. It'd be nice if they started to evolve antibiotic resistance at this point, but we haven't seen too much of them going back that way yet. All right, they do these crazy things. This is called horizontal gene transfer, or HGT. Different species can share genes. Can we share genes with different species? Not really, right? I mean, unless you had some kind of like laboratory intervention, we don't share genes. We can't just like go to, petting your dog. Your dog doesn't give you genetic material, but they can do this. So they can pass genes to different species, called horizontal gene transfer. Really crazy. Some eukaryotes can do this also, some of the smaller ones like some of the protists, but it's like really prominent in the prokaryotes. Okay, so how they do it. It's called conjugation. And here I say prokaryotic, I have in quotes mating, meaning it's like kind of like mating, it's an analogy. But it's not truly mating. Mating means that you use sperm and egg to make a new organism. But what happens is that these two, these bacilli bacteria, these rod-shaped bacteria here, they are not the same species. You can see this one has all these fuzzy things on them. These are called pili, some extensions that can come out from a prokaryote's body. And they'll do one of two things with these pili. This could be one of two things. One, it can be a special pili that can basically like poke into a different bacteria. Could be the same species, but here we can see they can do it with different species, and they can send genetic material across. Now, bacteria, prokaryotes in general, is they have one circular chromosome called a nucleoid. They've got this one main piece of DNA, it's called a nucleoid, it's a circular chromosome, but they also have other pieces of DNA that float around in their cytoplasm. is they also have these little plasmids that float around in the cytoplasm, and they also have DNA, which means they also have traits on there. And so it's these little plasmids that they can shoot across on what's called either a sex pili, or sometimes some of them can use a cytoplasmic bridge where they can take their cytoplasm and push it out through a pore in the cell wall and then connect the cytoplasm together between two different species, and then they can shoot those plasmids across.
Very romantic.
It's pretty wild. What'd you say?
Very romantic.
Yeah, yeah. So this is another way that they can evolve, is by sharing pieces of DNA, the plasmids, in this way. So now, because of this, we're seeing new genetic combinations in different species that you never see in one species. This is a big problem for us with antibiotics, because some genes for antibiotic resistance are found on these plasmids. And so now different species who are maybe present in a hospital, because a lot of people come in sick to a hospital and they have a lot of different kinds of bacterial infections. Plus, in general, we have our microbiome, all the microbes that live in and on us. And a hospital makes for a real good place for this to happen. Makes you think like, oh, should I go to the hospital? Well, yeah, because they can help you, but they have all kinds of new techniques that they're doing to try and combat this as well in the hospital. So what process are bacteria able to gain resistance to antibiotics? Which one? Okay. Oh, good. Conjugation. All right, this is important to understand. It's one of those sentences where you gotta maybe think about it for a minute. A pathogen, let me tell you pathogen definition. A pathogen is a disease-causing organism. I think I have it below. I think it's also, the definition might be right below this. So a pathogen can be a bacterium. Bacterium is single. Bacteria is plural. A pathogen can be a bacterium, but not all bacteria are pathogens. So not, here's one of the things in our societies that we often hear the term bacteria and you get like, ooh, bacteria, that's bad, right? But most bacteria are actually beneficial to us. There are very, very, very few bacteria that are pathogenic. So majority of your bacteria in the world are necessary, they're symbiotic. We rely on them. We have a whole ecosystem in and on our body of microbes, which include bacteria, but there's other things that live in and on us that help us. So this symbiotic relationship, meaning that we rely on them. They rely on us, they get a home. They get energy, warmth. It's just those few times you get that pathogenic one that can make you sick. About 1% of 1% of all the bacteria that are out there are actually pathogenic. So it's a really, 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 really small amount, tiny amount. So let's talk about the abundance of prokaryotes. So a drop of seawater has hundreds of thousands. A drop, hundreds of thousands of prokaryotes in them. A drop. A spoonful of soil, billions of bacteria, prokaryotes. The human body has trillions. They're all over. The human body, every every one of our cells, we have 10 microbes. Every one of our cells, we have 10 microbes for the relationship. This 1 to 10 difference, the microbes, they account for about 3 pounds of our body weight. So they're so little, they only account for about 3 pounds. All right, I'm going to talk a little bit about archaeas in particular, but not get into too much detail, but archaeans live in the most extreme environments. They're often called extremophiles. So hot springs. Hot springs, there are some hot springs that are actually boiling. You can find archaeans in there. We would dissolve in boiling water. You go down, deep into the ocean. There's a bunch of issues that would not allow most organisms to survive. One is water pressure, that as you go down in the ocean, the amount of weight of the water above you is crushing. It's heavy. I know like when you go in water, you feel weightless, but if you go underwater, it actually is like pushing on your body. If you go down thousands of feet in the ocean, thousands of meters, I should say, these bacteria here, there's shrimp, there's all kinds of crazy tube worms and things that live there. They can withstand that pressure, all that weight of the water above them. The other thing is that a lot of times there are fissures in the core of the earth that release boiling, hotter than boiling water. So they can live, there's communities that can live in more than boiling temperatures. And it also releases chemicals from the core of the earth that are poisonous. So imagine you can live in an environment where you're like constantly being kind of like smashed, it's poisonous and it's more than boiling. Thriving communities. They can live in the opposite direction, sea ice. So they can live in a frozen environment. They can live in hot salt concentrations and high salt concentrations. So, you know, most organisms, the salt would just suck all the water out of you, but they can survive in those. So those are the most extreme ones. Okay, so here's pathogen. A pathogen is a disease-causing organisms. They produce a set of symptoms based on some kind of like toxin that they release. So this is just a good reminder. I know when COVID hit, everyone's like, wash your hands for 20 seconds. And everyone's like, oh, we have to do that for, you know, the virus, get the virus off of us. But this is like normal. It's like what you should do all the time, even not when we're having a pandemic. Just good hygiene. Wash your hands for 20 seconds, just like brushing your teeth should be 2 minutes of brushing your teeth. Okay, so let's talk about some examples of pathogens. One is called Clostridium botulinum. It causes botulism. You ever go to the grocery store and there's a dented can? You don't want to buy that because it can have a little tiny like hole in it, and they thrive in that environment. What happens is that if you say, let's say that you have a can, good, it had a little like dent in it, and Clostridium botulinum is growing in that, and you eat it, what that bacteria does is it produces a toxin that paralyzes our cells. And so when you take that into your body, we're gonna find that the digestive system has all of these blood vessels that are taking food and nutrients out of our digestive system. So that toxin that's produced, it starts to paralyze your digestive system, but then it reaches into your circulatory system, and your circulatory system is in contact with every cell in your body. So your body starts to paralyze from the inside. Clostridium tetani, I'll just mention this one, I'll come back to that. Clostridium tetani causes tetanus. This is a lot more localized of that paralyzation. That like if you are walking around and you step on something that is rusty, there might be the Clostridium tetani on there, and then that area, that wound will paralyze. So it won't go to your whole body. Tetanus shots should be updated every 10 years. So if you don't know when your last tetanus shot was, you should think about that. I know mine is on the 9s. Every 9th year, I gotta renew my tetanus shot. So here's some interesting research that somebody went, oh, okay, so this bacteria causes paralyzation of cells. What if I take the toxin that Clostridium botulinum produces and I modify a little bit, and then we inject it into our faces, in the muscles, and we paralyze the muscles in our face so that when we smile, the muscles are paralyzed, and then you don't get the lines from smiling because you can't, because your face is paralyzed, and then you don't get wrinkles. That's called, what's it called? Botox. So that's where Botox came from. So there's like applications, you can think about that into the beauty industry too, for some of these. Right. How many of you have had a strep throat? Anybody susceptible to strep throats? That you get them? Oh, only a few of you, okay. So like this group A streptococcus, strep throat are part of that, but most of your common, your group A streptococcus are actually good and beneficial, but then some of us are just more susceptible to getting the sickness from this kind of strep throat bacteria. A lot of you who are not susceptible to the sickness carry it around in your throats. And when you cough and put it into the air and I breathe it in, I get those, and then it makes me have all these awful symptoms. So a lot of you are carriers to strep throat bacteria. Others in this class A of streptococcus are pneumonia, tooth decay. That's why you brush your teeth twice a day for 2 minutes each time. You want to make sure you get those tooth decay bacteria off. Rheumatic fever, cellulitis, toxic shock syndrome, glomerulophrenitis, and necrotizing fasciitis. So they go from like strep throat is not fun to like eating your flesh. There's a lot of diversity in here. These are easily spread from person to person. All right, so a little bit about necrotizing fasciitis. It is rare, but we hear about it every so often. The way that they can stop the infection, I know a lot of you are going into medical fields, is that massive antibiotic treatment, as well as they have to kill all of the bacteria. So depending on how quickly it is caught will depend on what they do in terms of treatment and surgery. One of the main things that they do is they just burn it. And so they use like hot tools to burn the tissue that's infected and hope that they kill the bacteria in doing so. Sometimes if it's really bad, they'll just like cut off your leg. Like they might go like, okay, it's all in the foot, we better cut off like maybe right below the knee. Or maybe it's gone pretty bad and they cut off the majority of your leg. But this one's a pretty serious one. Some bacteria are good. We're not talking about all, oh no, we're not talking about good ones yet. Sorry, we're still talking about bad ones. Biofilms. Biofilms are crazy. So bacteria have an easy time surviving, but they can go into these like thick, slimy layers called the biofilm, and they can survive in the harshest of environments, including like antibiotics, for example. Bacteria that can produce biofilms are really, really hard to destroy. Some, they believe antibiotic-resistant bacteria have the ability to do this. And so that's why antibiotics are really difficult in some cases to use. So check this out. Research shows that a species in a biofilm can be 500 to up to 5,000 times more antibiotic resistant than without a biofilm. That's terrifying. So go into research about biofilms, figure that out. The other thing is that they can live, a bunch of different species could just join together in one biofilm. So there's a lot of question of like, as the Arctic sea ice melts, Are there some in biofilms that have been sitting around for millions of years, and what are they going to do when they come out? So I'm giving you lots of kinds of nightmares today. Infections often develop gradually. You need intervention. You need some kind of like medical treatment, antibiotics, burning, whatever it is, to get them to kill off these kind of diseases. Sometimes people have chronic infections, and infection just comes back and comes back and comes back because they're like, wait, they have this bacterial infection and it goes away, but it goes away because the bacteria, some of them go into a biofilm inside your body. And then after you're done with the antibiotic treatment, they come back out again, and the infection blows up again, and then they go back in and out, in and out. So a lot of research needs to be done and is being done. All right, intestinal bacteria and foodborne illnesses. So good and bad. You've got a lot of, just like streptococcus, we've got a lot in our body that we need. There's some that are pathogenic, same thing with our intestines. A little bit about the microbiome. Microbiome is the environment, the ecosystem that lives in and on you. We are born sterile. You pick up your microbiome from your environment. A lot of it comes from the birth mom. Your microbiome is hugely influenced by who you live with and who you associate with. So that's why sometimes you see that groups of people are unhealthy because they all hang out together and they have unhealthy habits which produce an unhealthy microbiome. If you live in a house where you all eat pretty healthy, you might see that everybody's healthier. They often say that people who have pets, like dogs, they believe add very positive things to our microbiome. But here's the thing, a lot of research going on here too. A lot of opportunity for you all. There's so much research going on about the microbiome because they believe that diseases have two big parts, well, three big parts. One, genetic factors. Like, I haven't had anybody in my family who's ever had diabetes, so I don't believe that I have any genetic predisposition to getting diabetes. However, two, my environment, I could change that, right? I could get type 2 diabetes if I eat a high carbohydrate, sugar diet. I don't exercise a lot. I could, in my older years, I could get type 2 diabetes. But that's one thing that I try and control. And three is that maybe I control the, like I don't have the predisposition, I keep my environment pretty healthy, but I'm around some people who have diabetes, who have type 2 diabetes. I could pick up their microbiome and I could get type 2 diabetes there. They believe that our microbiome has a huge influence on whether you have diseases or not. So here's a weird thing. There's so many cool things to talk about, but okay, I'll just, let's get through this another day. Okay, so this is a very unfortunate name for something that is actually really useful. They're called fecal transplants. That insinuates that you're going to take somebody's feces and transplant it inside of you. That's not really what happens. What happens is they take feces from a healthy person who they don't believe has genetic predisposition to the disease you're suffering from. They have a healthy microbiome in terms of the disease that you're suffering from, and they take their poop and they put it in a centrifuge, which is a machine where you can spin solutions down, and the light stuff goes to the top and the heavy stuff goes to the bottom. The feces matter goes down and the microbiome floats to the top. And so then they micropipette off the microbiome. They might put it in like a suppository and then you gotta shove it up your butt, or they might put it in a pill and then you just take the pill. So it's not like technically they're transplanting feces, but it's the microbiome of the feces. There are companies that you can look up where if you are healthy and you're not predisposed to certain genetic diseases, you have a good healthy diet, you could sell your feces. I know you used to say like sell blood, sell plasma, but you poop every day. if you're healthy, you could make some money. Weird, but hey. Okay, E. coli. E. coli is the most common thing in our body, or most common in our digestive system, I should say. Most E. coli are really, really good for us. Some of them cause foodborne diseases. Where they are carried, the ones that cause foodborne diseases are in cattle. There are strains of E. coli. E. coli goes beyond like the genus and species name. They do what's called kind of like a subspecies. We call it a strain. They're different genetically, but they're still the same species. And some of these can infect cattle. And then the way that they do the factory farming is that cows come in, hung on a conveyor belt, upside down, and the first room, people just slit their throats and let the blood drain out. And then they go to the next room, and then there's a team of people who start cutting off the skin, and then they go to the next room, and then there's a team of people who might cut the rib meat off, and so then as they keep going, when they get to like ribs and the like butt material, it gets close to the digestive system, so occasionally people will like nick into the digestive system and get the foodborne E. coli onto your meat that you're going to eat. You eat that meat, maybe you don't cook it. If you cook it well enough, you won't suffer from this disease and get that intestinal bleeding, vomiting, diarrhea, dehydration, kidney failure. If you cook it, you don't get that. The USDA, it tests about 10% of our meat only. Let's see what's going to happen to that percent that gets tested now. There's a lot of restructuring of the government. Okay, let's talk about a couple good ones. I could talk a lot more about that. All right, nitrogen-fixing bacteria. These kind of bacteria live around the roots of plants. Okay, to put this in here. They can capture nitrogen. What we call this is fixing. This is really important to write down, just like going forward. When you hear the term fix, in biology, it means capture. So if you're a nitrogen-fixing bacteria, you can capture nitrogen from the atmosphere. We can't do that. We breathe nitrogen in, we breathe it out. We can't fix it. It doesn't go into our bodies. The way that we get nitrogen is by eating plants. You eat a plant, you eat something that ate a plant, you get your nitrogen. Nitrogen is a really important component of DNA, RNA, and proteins. 2 of our 4 big biomolecule groups. So nitrogen is super duper important. What if something happens to these bacteria? We'll talk more about that another day. Also, bacteria, like fungi, are a part of our decomposer group. There's a lot of bacteria in there. They can biodegrade. They can break down living things. They can make the nutrients available in our waste and in dead things back to the plants and the soil and the atmosphere, and then all that gets put back in through the soil and photosynthesis, and then we see the recycling of carbons, hydrogen, nitrogens, all of the atoms. There's a whole field called bioremediation, another really cool, amazing research field. They take living bacteria and they use them to break down waste environments, like toxic spills, like oil spills. There's been 2 really major oil spills in my lifetime. One was in like the 80s, and that was Exxon Valdez. And they used bioremediation to clean it up, and it's actually like really like healthy now. The one that happened in the Gulf about 8-10 years ago, the BP oil spill, they used what are called dispersants to do it. Dispersants take the oil and they make the oil slicks into little tiny, tiny pellets and then drop them to the bottom of the ocean. It disperses them, it doesn't get rid of them. Bioremediation is a much healthier way to do it. We'll see the effects of what happened in the BP oil spill and use of dispersants for tens of years. All right, what is the difference between a beneficial bacteria and a pathogenic bacteria? A pathogenic bacteria is
Capable of causing diseases.
capable of causing disease, all right. And, okay, one little bit. Almost done. All right, zoonotic diseases. Zoonotic diseases cross species barriers. So right now we're talking about the bird flu that in our neighborhood, a whole bunch of ducks were found along Lake Michigan, in Chicago as well as in Whiting, Indiana, very close to us, and they have the bird flu. They're also just found them in cows. It's bird flu. They also found one in a human. Bird flu. It's crossed the species barrier. It's a kind of virus that has crossed a species barrier. That usually doesn't happen. These are called zoonotic diseases. They can go from one species to another. Typically, we're most concerned with animals. Here's a whole bunch of zoonotic diseases. The coronaviruses, I know some of you are really familiar with MERS, SARS, COVID, right? They believe COVID came from a bat. West Nile virus, another one that you're probably familiar with. So we hear about these, the bird flu, the pig flu, swine flu, Lyme disease. Okay, so, ta-da! Lecture done. Okay, so how many pages you got? 97 pages. Hopefully you've been doing your objectives.